Do it.
Okay.
Good morning once again, Harley. It's great to be here with you again. Thanks for taking the time. It's always a pleasure to talk to you. Looking forward to it all week.
Okay. Yeah, me too. This is fun.
Yeah, we were talking about, we've been talking so much about Brix and uh, um, stress survival, stress management, and the sugar levels in plants. So we've had a lot of customers call in asking about that. What can I do to get my sugars up? How do I test the Brix? What do I need to know? And I've discovered, there's really not one answer. There's not
Right.
one answer to tell people. You know, you have to do this to make it work. And so it's more of a combination of everything. I like to tell people it's a great balance between nutrients, sunlight, um, you know, temperatures, the just growing environments. There's so many things involved there. But the first thing we try to direct our questions or direct our customers to do is tell them that um, one thing we like to do is minimize the amount of nitrate that we use. That's step one that I always tell them. Excess
Right.
nitrate leads to a lot of problems, and it's the first go-to for everything. Put more nitrogen on it, and so on and so forth. And we try to tell them it's not an excess nutrient that pushes yields or controls quality. It's a lot of times the ones that are missing that control it and being out of
Mm.
balance. So that's what we kind of instruct them to do. But everybody's garden's different. Everybody's regimen is a little bit different. So it's just a combination of many things.
hmm Yeah, I agree. That's, nitrogen is number one. It, Nitrogen is a luxury element. If you give the plant the nitrogen, it will take it up, whether it needs it or not. You give it more nitrogen, it stimulates the plant to take up more nitrogen. But it's energy. It's done in the chloroplast. It takes a lot of energy to assimilate nitrate nitrogen into changing it into amino acids and proteins. So maybe as much as 30% of the energy of photosynthesis just goes to assimilate the nitrates. So you overdo it with nitrates, it's going to lower the Brix, it's going to lower the stored energy in the sap, and it's just going to burn it up as top growth. And that's not necessarily good. Your quality goes down, and also you don't have extra sugars to send to the roots. So literally, you overdo it with nitrates. Oh, look how big my plants are growing, but the roots are restricted, and you need the roots to take up the nutrients to make the sugars. To improve the Brix. So yeah, you're right. Nitrate, having it at the high end of sufficiency would be great for production, but not in the luxury levels. The other one I look at is potassium. Um, if you want to adjust the nitrogen to potassium ratio. If your Brix is a little low, you can either lower your nitrates or you can raise your potassium, because that is responsible for carbohydrate metabolism and also for shipping the sugars through the plant. Um, so you either lower the nitrogen, raise the potassium, or compromise, do a little of both until your Brix starts to go up.
Oh, gotcha. That's great information. Yeah, I agree with you too. And one thing that we like to instruct a lot of our customers that call in or message us is let's take a look at all the forms of nitrate that we're using and what can we eliminate nitrate wise and see if we can't find a sulfur or a sulfate wise. So like a big one's, potassium nitrate. Everybody uses that. Calcium nitrate. Everybody uses that, and it's much needed. And
Mm-hmm.
then magnesium nitrate, and so we're
Mm-hmm.
like, let's supplement some of these nitrates out for a sulfur form, and that really helps the plant build proteins. Just having that additional sulfur and the reduction of actual nitrate in the system. And for a soil grower too, we try to convey to them, let's find other forms of nitrogen to use, more protein type nitrogen or amino acid type or things of that nature, some slow release things in the soil, because the ammonia aspect of that too really helps produce you know flowers and fruits and things like that, where nitrates can get in the road sometimes.
Yeah, yeah, I can see that especially in soil. But in hydroponics, I like to give it about 90% of the nitrogen in the nitrate form, and no more than 10% or so in the ammonium form. But in soil, you're feeding the microorganisms.
Mhm.
So they can have a higher ammonium to nitrate ratio because that's how they do it in nature. The microorganisms make digestive enzymes. And one of those enzymes is called a protease. And it breaks down proteins into the building blocks of proteins, which are amino acids. So they break them down, and then they use the amino acids. They'll snip some of that ammonium off of there for their nitrogen needs. Um, and also, there's some they'll change that into ammonium, and then there's other microbes that change ammonium into nitrite, and other microbes that change nitrite into nitrates, and the plant takes them up. So in nature, you can give them a little more ammonium to start with because they're going to build up change it into more usable forms. And
Yeah,
they're doing it in real time right on the roots. So,
great, great
it is slow
information.
release nitrogen. Yeah, it is slow release nitrogen in nature. And the microbes, another cool thing. I'm the hydroponics guy, but I get real excited about soil because microorganisms, the bacteria themselves, rarely die of old age. They're eaten by protozoa, and then the protozoa use the nitrogen that's in the bacteria for their nitrogen needs, and then they spit out the excess, and that's plant available nitrogen again. But it's right there in the rhizosphere, right in the root zone, more real time. So it's like 95% efficient in nitrogen recycling in nature because nitrogen is so limited. Soil without organic matter, there's no mineral that has nitrogen in it.
That's
So,
true.
you know,
That's true.
they've evolved. Plants have evolved to be able to recycle the nitrogen that's available, and I think that's why it's a luxury element. If it's available, they go, "Oh, it's available right now. I better take it now while I have it," and then build it up. But again, it will end up lowering the Brix if you give it too much.
Oh, yeah. So glad you're the hydro master, and it's good. This is a nice combination of conversation we have between the two. It's very fun, very fun. For some of our organic farmers and some of our soil growers, uh, I can't remember how many tons of nitrogen is in the air per acre. It's something that's thrown around all the time. And just because it's there doesn't mean the plant can get it. So
No, you
we've done
can't.
some really interesting projects on growing our own nitrogen. So cover cropping the soil with
Mm-hmm.
clovers and alfalfas and things like that, and cutting it, letting it lay, and turning it into the ground. And it's hard to do as a farm because there's no profitability there. Basically, you're growing your nitrogen. Let's say they do cover crops for a year or two and keep turning it in, and then come in on year 3 and plant corn and have a bumper crop. And they set up the system and grew their own nitrogen. It can be done, but again, you're dealing with soil. It's a long process, and that goes along with what you just said. Without the addition of organic matter in the soil, it will never get mineralized into energy, through the
Mm-hmm.
biology and things like that. So, yeah, very good uh, ways of thinking. People can really consider this information and apply that to what suits them best.
Yeah, and I like what you're saying too about growing things like soybeans and alfalfa out in the soil. Well, the soybeans have microorganisms on the roots that in a symbiotic relationship, and I'm sure you know this, that literally fix nitrogen. They take nitrogen out of the air and change it into ammonium fertilizer right in the roots. And the roots actually make nodules for homes to protect the nitrifying rhizobium, they call them. Um, because that fixation of the nitrogen is done under anaerobic conditions. So they literally protect the plant from the oxygen so that they're able to fix more nitrogen. But the microbes will change it into the ammonium form of nitrogen, not the nitrate. Now, once the process is done, though, and more nitrates are produced in nature, they're very water soluble. You get a good rain, it washes the nitrogen away from the roots. And that's why you start to see nitrogen deficiencies a lot in organic growing. I've seen it out in the fields in Oregon. You get some heavy rains, they'll wash that away, and then the plant doesn't have enough nitrogen. Yeah, so again, though, the microbes are important for that. And Brandon, there's some microorganisms that literally increase the Brix of plants, not because of the nitrogen, and they might be part of it, but they do it but in other ways. One is some microorganisms like Bacillus subtilis GBO3. It literally makes volatile organic compounds. So if the microbe, even if it doesn't touch the root, it's not in contact, those volatile compounds can trigger up to 600 genes in the plant. And one of those genes tells the plant, take up more iron. Iron is a catalyst to make chlorophyll, to harvest the sunlight. Another one of those compounds literally turns off the sugar signal in the plant. When the plant gets enough stored sugars, and gets close to the end of its life, it says, okay, I don't need to do photosynthesis anymore. I'll just use the stored sugars I have left. But that if that microorganism turns off the sugar signal, the plant doesn't know it has enough sugar. So it continues to do photosynthesis, continues to build up the Brix all the way to the day of harvest. So nature has a way of stimulating the more production of the sugars from the plant. I'll ask you the question, this is kind of rhetorical, why would a microorganism make root stimulants? Why would the microorganism tell the plant to continue to make sugars? What benefit is it to the microbe?
It goes back to the symbiotic relationships and the biodiversity of the total ecosystem.
Exactly. Every cell in every living organism on the planet, the genetic code says the same thing. Be fruitful and multiply. So the microorganisms, if they can stimulate more root mass, they colonize the roots. And as the roots are growing, the colonies can grow with it. Be fruitful and multiply and take over this perfect environment. Also, if they stimulate the plant to make more sugars, some of those excess sugars are sent to the roots and leak from the roots to feed the microorganisms. So their metabolism is improved, and then they start to divide. And guess what? While they're dividing, that's when they make the enzymes, and that's where they make the biostimulants. So it's this beautiful cycle of nature. The plants are feeding the microbes are improving the uptake of nutrients and stimulating the production of sugars for the plant, and also to keep that whole system going.
Yeah, in our minds, we got to keep a clear picture of the root system in our head when we're looking at our gardens, because we're always looking at what's up top. We don't think about the root system. But anyway, we have a great product. It's called Route 66. It's a new
Yeah.
inoculant. We've had it for about a year, and it has some of the highest concentrations and some of the most species all blended together. It's very effective. But I just wanted to show that where we were talking about the roots. I happen to have a bag right here on my desk.
Okay. Hey, let me see let me try something here we haven't done before. I'm going to try to pull up a picture for a minute. Let's see. I go to share. Okay, wait a minute. First thing I'll do is I did this morning because I wanted to see the picture of the Brix refractometer.
Okay.
So let me go here, share my screen, share screen, and go to applications, do this, and share. Okay. Can you see that now?
Oh, perfect. Yes, that's a great, yeah.
Okay, I'm going to
There it
pull
is, the
this one.
refractometer.
Yeah, I'll pull this. I'm going to move this out of the way slightly. Slideshow from current slide. Okay, this is a slide that was from my class, one of the classes I do in the Master Grower Short Course, and it's on microbial inoculants and compost teas. So there's just one slide there about increasing Brix. Brix is a measurement of the sugar content in the sap. It's done with a Brix refractometer. Here's the Brix refractometer. What you do, and you know this probably better than I do, you take a fully developed leaf near the top of the plant that's in the light, take that off, you squeeze one or two drops of sap onto this slide here, right there. You close the cover, then you look through it at a light source, like you'd be looking through a telescope, and you can read the Brix level just like it was a thermometer. You can read the different levels of Brix. They also have digital refractometers that will give you the Brix as a number, as digitally, instead of looking at it like this. I like, personally, for my own use, I like the digital one in the lab, because then I have a real number to write down. But I like this manual one in my garden, because what I really want to see is a high Brix, but not a real sharp line of demarcation, kind of a fuzzy line, because then I'm reading not just sugar level, I'm reading total dissolved solids, which will show me vitamins and organic acids and amino acids, other things that are there. So if I get a high Brix with a fuzzy line of demarcation, that's the best for me in my garden. But in the lab, though, I don't want to kind of guess, where is it? It's between here and here. I want an actual number to write down to make sure I'm accurately reading increases in Brix. So some of these microbes increase the photosynthesis process itself. Like I said, the volatile organic compounds stimulate responses in the plants, activate genes, upregulate the iron uptake. So some of the microbes literally make the iron more available. They make siderophores, iron carriers. They make other kind of Krebs cycle intermediates that chelate iron. But then they literally tell the plant, change the homeostasis of the plant and say, take up more iron. Take up more iron, they make more chlorophyll. And then one of those compounds also down-regulate the sugar sensing. So the plant continues to build up sugars over time. And it improves the flowering process too. And even early on, not just at the end during flowering, early on, the excess sugars that the plant makes are shipped to the roots and they're stored as starches. When the plant transitions to flowering, it uses that stored energy to take up phosphorus, to take up potassium. And phosphorus stimulates earlier flowering and more flowering sites. So I found when I was using the biostimulants with the right level of nitrogen and potassium, I saw 20% more flowers in the early flowering stage, the transition from grow to bloom. Even though it was a quote unquote grow formula, it isn't just giving it more phosphorus or potassium, it's the plant taking up the phosphorus and potassium. And so sugar levels in the roots were a key to that process, improving that process. So I'm going to take this back down. I just wanted to share that with you because you wanted to see the picture
Yeah.
of a Brix refractometer.
Yeah, that'll help some of our questions and customers that have been asking about that. So that's a good display. And I like what you were saying about um, uh, the fuzziness of the line. A lot of times there is not a definite line. Sometimes it's so thick you really have to pinpoint, uh, you know, what your parameter is. So I like to tell people if they're new to testing Brix and the sugar, put a drop of a soda on it, put Mountain Dew on it, and you can really see what that line is. And that's not what you want to see from a plant because, like what you said, you want a full display of the sap in there. And so that's very well said. That's important for a lot of people to understand. It's not going to be a straight line. You don't want it to be a straight line. You want it to kind of blend together and look for where it contrasts together.
Mm-hmm.
Good point.
Exactly. If you see a straight line, you're just looking at sugar, but there's more going on there that shows the health of the plant. Again, you know what, I don't know, maybe we talked about this too, but in my own garden sometimes, if I don't even use the Brix refractometer.
Yeah, you just taste it.
Did I tell you that before? I just
Yeah.
pop a leaf and touch the sap to the tip of my tongue. And if
Mm-hmm.
it's sweet, I know everything's in balance. If it's a little astringent, a little acidic, puckering up a little bit, then I'll make my adjustments to either potassium nitrogen ratio or even add some biostimulants to aid with uptake of nutrients.
Okay, good. So let's just recap. Don't overload on one nutrient expecting high Brix. Try to keep the balance of minerals. Um, the plant needs a good environment too to build the chlorophyll and create the sugars. And then one question to a lot of inexperienced customers, they always ask, why can't I just feed my plant sugar water? Shouldn't that raise the Brix? I'm giving my plant straight sugar, and it's an easy answer for us to tell them is, plants don't drink sugar, they produce it. So be very careful. Everybody likes to grab that molasses jug or, do whatever. And again, I've seen more harm done by over applications of molasses many more times than I've seen the benefits of it.
Mm-hmm. Yeah, I definitely agree. Um, if you overdo it with the sugars, especially early on in the growth, it, microorganisms need carbon and they need nitrogen. If there's a bloom of microbes because you're giving it too much sugar, it will literally compete with the plant for nitrogen, and then you can get a nitrogen deficiency. You don't want that. I have to be careful because I'm always preaching against don't use too much nitrogen. And then people think,
Mm-hmm.
oh, nitrogen's bad. No, it's not bad. They need nitrogen more than any other element. But you don't want a deficiency, and that can happen. I've seen it in the lab. I did a test in hydroponics years ago. We set up in a warehouse, set up a whole bunch of tents, and we each one had a controlled environment, each had a hydroponic system, and I was testing the sweet products that were on the market. Some of them had molasses, some said sugar. They had different kinds of sugars, carbohydrates. So what I would do is I'd mix up a nutrient solution, full strength, with all my nitrates, everything in it. Right then, before I put plants in, I would add the sugar product as directed on the bottle. Whenever it said, okay, add that amount, mix it up, took a nutrient sample right then, immediately, sent it to the lab. Sometimes 3 days later, when I got the response, got it back, 0 nitrates.
Oh, wow.
What? 0 nitrates? How is that? I know there's got to be a mistake. I told him, you must have made a mistake at the lab. I put that in myself. I know there's got to be, that was really strong. I must have had 250, 300 parts per million nitrates, on that full strength. 0 came back. Had them retest. It was 0. And I said, what is going on? There are some microbes that are denitrifiers. They actually convert nitrogen back into nitrites and then down into final ammonia and then off gases. That's just part of the nitrogen cycle, part of nature. Well, in hydroponics, I had 90% of the nitrogen was nitrate and only a little bit of ammonium. So I was giving a competitive advantage when I had the sugar to the microbes that fed on the nitrates and turned them into nitrites. And nitrites are toxic to plants, even in small quantities. So I assumed it was nitrates, but then I got some chemicals. I did some titrations, and lo and behold, did the measurements. The nitrates were converted into nitrites. So every single system that had sugars added performed worse than the control with no sugars. Even with, with the lab test with the leaf, Brix, everything was lower. And you could even tell just by looking at them that these plants don't look as healthy, and some were looking really bad. Now, on the other hand, with the amino acids, it didn't do that. Didn't have that negative effect. It was stable. It stabilized because the amino acids were converted into ammonium, and then the ammonia nitrates, nitrates. So it was a nice stabilizing effect. In aquaponics, that's a good source of nitrogen, is a stable form of nitrogen for your system, because it feeds the bacteria in a slow release way in the biofilters to keep the water clean and not get too much ammonium build up over time. So yeah, I did the experiment. In the end, we said, let's don't make this product, this sugar product. Now, I will say this one thing, Brandon. I know I'm kind of monopolizing our talk today. I'm sorry about that.
This is
But
great info, though. Keep it up.
yeah, yeah. But at the end of the process, toward the end of flowering, or even if you're doing some flushing in the hydroponics, the sugars I don't think are a bad idea there, because the plant's already stored up all the nitrogen it needs. The first half of the plant's growth, it's simulating about 80% of the nitrogen it needs for the whole life of the plant. And it's okay to bring them down some of that nitrogen at the end, because that nitrogen that's not assimilated can add off flavors. And also at the end, plants stop leaking sugars because they want to hoard their sugars for the seeds and the fruits for the next generation. So they don't feed the microbes anymore. The microbes in the roots start to go dormant. They go to sleep. So at that point, when the plants stop feeding them, maybe it's a good idea for us to feed them, because we don't have next year. We're going to be harvesting and starting over. Nature, it's a good way to do it. But but giving them a little bit of the molasses at that point, the microbes will feed on that. They'll stay active, and there's iron in the molasses too. That's good. So the plants will make that iron available. They'll turn off the sugar signal. The plants will take it up. The buds or the fruit could sweeten up all the way to the day of harvest. So there is a place for it, but definitely don't overuse it, and don't use it too early in the crop cycle, in my opinion, from what I've seen with the data.
Yeah, once again, great information. Great information. That is so helpful to a lot of people. They just, a lot of our customers get sidetracked off on something they read on the internet. And all the points you just made goes back to just a common theme is don't overdo it on any one thing. Again, it's a balance of nutrients. Nutrients in excess, or sugar or molasses in excess, anything in excess will not drive yields. That will not drive quality. It's back to that balance, and a combination of those balances, and just such a great point on molasses. That's going to help a lot of people. I'm glad we had this discussion.
Yeah, me too. Maybe next week, and for another topic, I'd like to learn more, talk a little bit about, but especially get your input, a little bit more on compost tea. Just
Oh boy.
Because you add molasses to your compost tea to feed the microbes, right, or something like it. Maybe we can talk more about the inoculants and the compost teas next week, because I bet you have a lot of good stuff to share.
Oh, we sure do. We sure do. That'd be great if I could show some pictures too, because we have 10 compost. Let me back up a little bit. It's referred to as compost tea, but you know what? We don't use any compost in our compost tea. We like to inoculate it with other things, but that's just a term of the industry, compost tea.
Okay. Yeah. So let's put that on the shelf. I think we're out of time for today, but maybe we can put that on the shelf and bring that up next week. I'd love to see some of your pictures and hear your input. I have a little bit to share on it, but between the two of us, I think it'll be a good but a good discussion.
Oh yeah, this will be fun. Oh yeah, this is I'll get my information all put together, and I could send it to you so you could be able to share it in the format that kind of what you've been doing with the slides. Maybe you could arrange it in that, and we can have some great discussions and show you a lot of tools we use, how we do it, why we do it, the end results we're looking for. Oh, that'll be fun. I'm looking forward to it.
Yeah, it's going to be the perfect progression too. Oh, one last thing I wanted to say about this though. Remember we talked about the iron uptake for the for making as a catalyst to make the chlorophyll. a lot of the agronomists out there, when it comes to molasses, they attribute most of the benefits of the cane molasses not to the carbohydrates, but to the iron and the iron availability. Just wanted to throw that out there at the end.
No, that's true. And there's some available, potassium in it too.
Mm-hmm.
Yeah, but yeah, the
Okay.
iron, I agree. Yeah.
Okay. Well, thanks for inviting me again. I love doing this with you. I've got one little news break. This week, my website is going to go live, and it's hydroharley.com. And I already have our webcast. The last two webcasts are already on that site to view. So it's just making it go live, and that should be any day now. So just
Hydro
throw that out
Harley.
there.
Okay. Good announcement
Hydroharley.com. Easy to remember.
from the man himself. Looking forward to it. Yeah, we'll support that as much as we can. That'll be great.
Okay. Oh man, I can hardly wait till next week already.
Yeah, we'll do it. It'll be a good one. Everybody's got to join in and be ready for that one. It's going to be good. So, okay, well, have yourself a good rest of the week. I'll do the same. And yeah, we'll get back to work. Looking forward to next week.
Okay. See you later, Brandon.
All right, take care, my friend. See you. Thank you.